Recall that nervous tissue is dominated by two cell types. The first cell type was the neuron, which was the structural unit of the nervous system, and the neurons were the signal-sending cells of the nervous system. So they created electrochemical signals, they created the gradient potentials, the action potentials, and they also released neurotransmitter. And their job was to, or is to, signal other cells and tissues and control homeostasis or maintain homeostasis. On the other hand, we had the neuroglial cells or the glial cells, and these are the supporting cells of the neurons and nervous tissue. So they help to maintain neurons in various ways because the neurons are very highly specialized for sending signals, and so the neurons are not very good at sort of the housekeeping functions of the nervous system tissue. And this is really what neuroglial cells do, is they perform a lot of the basic maintenance and housekeeping functions of the nervous system tissue. So here we're going to focus on the types of neuroglia and their specific functions. So in general, the neuroglia or the glial cells are non-excitable cells, meaning that a stimulus will not change their membrane potential, and it's not going to cause the release of neurotransmitters because neuroglia don't manufacture or release neurotransmitters. This is in contrast to the neurons, which are excitable cells that release neurotransmitters to signal other cells or tissues. So in short here, what we're saying is that the neuroglia are unable to send signals like the neurons are. Neuroglia are found in both the central and peripheral nervous systems. There's a couple of different types of neuroglia within these anatomical divisions of the nervous system, and we'll look at their specific, those specific cell types and their functions as well. So instead of signaling, the neuroglia support and maintain the environment around the neurons, and they also perform a lot of the maintenance and housekeeping functions within the nervous system. So let's start with the neuroglia of the central nervous system. So these are going to be the neuroglia that are found within the brain or within the spinal cord. And there are four types of neuroglia that are found within the central nervous system. There are the oligodendrocytes, the astrocytes, the microglial cells, and the ependymal cells. So let's start with the oligodendrocytes. So oligodendrocytes are cells that wrap the axons of nearby neurons, and they create a covering over that little part of the axon, and that covering is called myelin. Now, what myelin helps to do is it helps to increase action potential speed, so that as action potentials traveling along this part of the neuron, or this part of the axon, I should say, will actually travel much faster if myelin is present, and so this helps to increase the speed at which neurons are able to signal other cells. So those are the oligodendrocytes. Let's take a look at the astrocytes next. Now the astrocytes are named because they are star-shaped, because astro means star, and cyte of course means cell. So these are the star cells. And the astrocytes have being star-shaped, they have a number of these branch-like processes that come off of or emerge from the cell body. And these branches reach out and cling to nearby neurons. They also cling to nearby capillaries and blood vessels as well. And it turns out that one of their functions is to help facilitate the exchange of nutrients between the capillaries and the neurons. One of the things that they also do is they help kind of clean up the environment around the neurons. Specifically, they're cleaning up any excess neurotransmitters that remain in the space between neurons after neurons release them. And they also have to clean up excess potassium ions as well. So we see that they're kind of performing a cleanup function of the chemical messengers as well as some of the ions in the spaces between neurons. Another thing that astrocytes also do is they control the formation of tight junctions in the blood vessels of the brain. And so what you're seeing in this diagram here is you're seeing kind of the basic structure of most capillaries within the body. And most capillaries in the body are actually quite leaky. That is, the epithelial cells, or they call them the endothelial cells, but this is actually, it's an epithelium because it lines an internal cavity here. But these epithelial cells have little gaps between them. They're called intercellular clefts. And this means that fluid like blood plasma and any chemicals or ions can usually squeeze between the cells that make up the walls of the blood vessel and get into the tissue around it. So that's kind of the basic structure of most capillaries in the body. Now, capillaries in the brain are much more secured. They're not as leaky. And the reason for that is that the endothelial cells are joined by tight junctions. And so what this means is that the capillaries of the brain are less permeable than typical capillaries. And the reason why these tight junctions are here is because the astrocytes actually stimulate or control the formation of these tight junctions in the capillaries of the brain. And what this essentially establishes something called the blood-brain barrier. So what it basically means is that it makes it more difficult, the blood-brain barrier makes it more difficult for substances within the blood to leave the bloodstream and enter the nervous system tissue. So essentially these brain capillaries are less leaky or less permeable than typical capillaries in the body. Really the one major thing that can get across the capillary walls of brain capillaries, and the same goes for most other capillaries in the body as well, are lipid-soluble substances, because lipid-soluble substances can move from the blood across the plasma membrane of the capillary cells and into the tissue surrounding that blood vessel. In this case, this would be the brain tissue. So one of the things that astrocytes do is they stimulate the formation and maintenance of these tight junctions within the brain. So essentially these astrocytes are kind of stimulating the capillaries to be less leaky than typical capillaries within the body. So that summarizes the functions of astrocytes. Let's move on to the microglial cells. Microglial cells function in a capacity that's very similar to cells of the immune system. So they break down dead or dying neurons in the central nervous system, and they also function to kill microorganisms like bacteria or fungi that enter the nervous system tissue. Because, as we saw in the previous slide, the blood vessels, particularly the capillaries within the central nervous system, are really not very permeable, and that means that the white blood cells that are in those capillaries have a hard time getting into the nervous system tissue. And so this means that even our own immune system has very limited access to the tissue of the brain and the spinal cord. But fortunately, there are these microglial cells that perform those immune system roles and help to break down dead tissue as well as kill any foreign microorganisms that are there. Last but not least, the last of our central nervous system neuroglial cells are the ependymal cells. And the ependymal cells are cells that line cavities within the brain called ventricles. And these are small cavities that are filled with fluid. The fluid is called cerebrospinal fluid, and this is a liquid that really helps to kind of act to support and cushion the very soft and delicate brain tissue. Cerebrospinal fluid is also found within the center of the spinal cord, as well as there's a thin channel that runs down the center of the spinal cord. So we see cerebrospinal fluid there as well. So the role of these ependymal cells that line the walls of these cerebrospinal fluid filled cavities is that the ependymal cells help to move the cerebrospinal fluid through the ventricles and within the ventricles. And the reason that they're able to do that is because they have cilia on their surface. So recall that cilia were these extensions of the cell membrane that were capable of movement, and the cilia wave and they beat and they move, and so that helps to actually kind of propel the fluid, the cerebrospinal fluid that's within the ventricles of the brain. So that finishes up our discussion of the neuroglia of the central nervous system. Let's briefly turn our attention to the neuroglia of the peripheral nervous system. There are two types of neuroglia that are found within the peripheral nervous system. They are the satellite cells, and there's another cell type called the Schwann cell. We can see both of those cell types in this diagram here. The satellite cells are here that are in purple, kind of surrounding the soma, or the cell body of this neuron here, and the Schwann cells we see as these little blue bubbles that are surrounding the axon of this neuron here. So satellite cells here function very similar to astrocytes, and so they help facilitate the exchange of nutrients between this neuron as well as nearby capillaries. The Schwann cells that you see wrapping the axon, you may have guessed just because they're wrapping the axon that they function very similar to the oligodendrocytes, and if you had guessed that, you are correct, because they do, and the Schwann cells form the same myelin sheath that's found in the neurons of the central nervous system. And so what Schwann cells do is they wrap nearby neuron axons and function as myelin in the peripheral nervous system. And remember that myelin is a structure that helps increase action potential speed. So action potentials that are traveling along this axon will travel at a much higher speed thanks to the myelin that's found around this cell.